In this video, we're going to look at some glycoproteins. So glycoproteins are proteins that have carbohydrates on them, and they can vary quite a bit. For example, a glycoprotein's weight could be anywhere between 1 and 9% from the sugars. And the carbohydrates on a glycoprotein have what we call a microheterogeneity. That is, it's very variable composition based on, you could have two proteins, the same amino acid sequence, and they would have different carbohydrates. That's what we mean by microheterogeneity. Or on the same protein, you would have different sugars. And glycomics is the study of these carbohydrates. And a proteoglycan is a protein plus a glycoaminoglycan that's in your extracellular matrix. So in the last video, we talked about hyaluronate. So hyaluronate is our big carbohydrate here. And off of hyaluronate, we have our proteins, which we call our core protein. And then on our core protein, we have other sugars attached. We have keratin and chondroitin. Now these proteoglycans have these sugars attached to the protein, and so sugars can attach to proteins either through an N-link or an O-link. This N and O is just telling you what atom you're attaching to. N-linked is through asparagine, while O-link will be serine or threonine. Now these hyaluronates can vary between 4,000 to 40,000 angstroms, so they can be quite long, and they can have up to 100 different core proteins. And each core protein can have 50 keratin sulfate chains and 100 chondroitin sulfate chains. This means you have a lot of alcohol groups, a lot of negative charges around, which means these will be just full of water, completely hydrated. And so these compounds make up your cartilage. So let's take a look at your cartilage here. We have your hyaluronate. We have your core protein and your sugars, which here we're calling an aggrecan. And throughout here we just have collagen all weaved in. This is just surrounded by water, making this nice gel in cartilage. That's why cartilage is flexible, because you have just sugar, water, and protein, so you can move this around some. Now, when we think about proteoglycans, we also see this in the bacteria cell wall. And bacteria will be either gram-positive or gram-negative. And this is just from whether they will take up a reagent we call the gram stain. Gram-positive will take up the reagent, gram-negative will not. So gram-positive have this thick peptidoglycan layer, which we'll talk about in a minute. Gram-negative has a thin peptidoglycan layer, but an extra outer membrane. And this outer membrane is what excludes any harmful chemicals like the gram stain. And so gram-negative bacteria are resistant to antibiotics for the most part because they can never penetrate the outer membrane and they cannot get to inside the bacteria to kill it. So your harmful bacteria are your gram-negative ones. But let's talk about this peptidoglycan. So peptidoglycan has alternating beta-1,4 linked glucanac and N-acetylmuronic acid. So glucanac is just a sugar, and N-acetylmuronic acid as shown here. Now this acetylmuric acid will form an amide bond with a D amino acid tetrapeptide. So here we have our muronic acid. Here is our tetrapeptide. In reality, it can range, it can be 4, it can be 5, it can be 3. So the peptide can change quite a bit, but it's alanine. Then we have isoglutamyl, which is glutamic acid just in a bond with L-lysine here. So the glutamic acid side chain is actually part of the peptide bond. And then we have D-alanine, one of our only D amino acids in nature. And so what happens is that these sugars are forming bonds with this peptide, which helps to cross-link the sugar molecules. And what you get is you get a net. So the peptidoglycan is really just a sugar and protein net. And these cross-linkings are completely random. 40% of the available cross-links are made, and you get holes of varying sizes. Well, one way to destroy the peptidoglycan and to destroy the bacteria, because they need the peptidoglycan to survive, is through an enzyme called lysozyme. Lysozyme will hydrolyze this beta-1,4 linkage here between muramic acid and acetylglucosamine. And lysozyme is found in your tears, your mucus, and other secretions. So every time you cry, you're crying out some lysozyme. And any bacteria that comes into contact with your tears or your mucus, they will die because they will lose their peptidoglycan. And penicillin, which is the first antibiotic ever found, well, what that does is it inactivates the enzyme that forms this cross-link. You don't have this cross-link, you don't have a peptidoglycan, and without a peptidoglycan, bacteria cannot survive. And that's it for this video. I will see you in the next one.